Hello students and welcome back to our channel. I hope that all healthy and being staying safe at home. So previously on the channel we have posted about a video on the cell. Okay, so today's topic is also a kind of a connect to the previous topic. So let's start with our today's video and the topic. So what we can see here in the image, we can see here there are two different types of cell, that is animal cell and the plant cell. Okay, about the different kingdom and each organ of the cell, which is called as organelle, we will do that in detail. Don't worry about it. But let's just focus here on the animal cell and the plant cell. Okay, now students, most of the animal cell and the plant cell, they are eukaryotic cell. Yes, they have a confined nucleus in which the DNA or the genetic material is present inside. Now, as we can see here, that the cell, each and every cell, whether it is a prokaryotic cell or a eukaryotic cell, they have a lining, they have a boundary, and this lining and boundary is nothing but it is called as cell membrane or plasma membrane, or it is also termed as plasma lemma. So today's topic is most of the connectivity with the 11th and the 12th standard biology. I will be discussing few of the important questions in detail which can be asked in your entrance exam. So let's move forward with the today's topic in detail, that is cell membrane. Yes. Now, why we have to study about the cell membrane? Because whether it is a prokaryotic cell or eukaryotic cell, they have the boundary, and this boundary is termed as cell membrane. In the case of plant cell, apart from cell membrane, they have one more lining which is called as cell wall, and some of the bacterial cell do have it, but we will be studying here most of the cell membrane. So how the cell membrane actually got discovered? So there were many scientists who have actually put a lot of effort to discover it and to actually come up with a model. So let's discuss the journey of the discovery of cell membrane. So the very first attempt was given by Davson-Danielli model in the year 1935. Oh wow, what we can see here? We can see here a sandwich. Yes, the name of the model was also sandwich model, where the Davison and Danielli, they suggested and they said that the stuffing which is there in the sandwich, the same thing is there in the cell. This stuffing inside the sandwich is nothing but it is a lipid, which is nothing but they are fats. And the upper layer, which you can see the purple color, is the protein, which is nothing but this is the bread. But later, due to some of the things which were not being able to prove by them, this model, which was given by Davson-Danielli, got rejected. And students, the thickness, okay, of the cell membrane was given by them was 7 nanometer, which was one of the good point. Now, the next effort which was put by were the Robertson unit membrane model. Yes, he suggested that the whole membrane is a one unit. It is not separately the protein and the lipid. It is all together. The protein and lipid, they are together. He didn't explain that there are two different layers. Later he said that there are two different layers of lipid, but the protein and lipid, they are together. But he was not been able to explain the further reasons for it. And his model also got rejected. Then the next attempt was given by the Singer and the Nicholson, and that was in the year 1972. That model was named as fluid mosaic model. Yes, student. What is this fluid mosaic model? Don't worry, we'll be doing that in our today's video. So the fluid mosaic model of the cell membrane was given by Singer and Nicholson. Yes. Moving further. Now, the cell membrane is made up of 4 major ingredients. The way the recipe is made up of ingredients, same way the cell membrane is also made up of 4 ingredients, or the chemical makeup we can say. So, the cell membrane is made up of lipids, proteins, carbohydrates, and cholesterol. Yes. As we can see, the cell membrane itself is made up of these three very important molecules. These lifeless molecules actually make a cell membrane, which is a part of cell, which is a living cell. And therefore, when we studied biomolecules, we have to study carbohydrate, proteins, and lipids in detail. Yes, a small part of it is also contributed by cholesterol, and therefore we cannot completely avoid having fats from our diet, because the cell membrane is made up of lipids and cholesterol. Okay, good fact. Okay, moving further, that how many percentage does each lipid or protein and carbohydrate, the ingredient, contributes to? So the lipid contributes about 40%, protein contributes around 52 to 58%, whereas 52 percentage of protein is present only in red blood cells. The carbohydrate is present only 1 to 2%. Okay, and cholesterol, we will discuss in detail where it is present and how much. Don't worry about it. Moving further, now it has also been suggested that the protein act as an iceberg. It is present in the cell membrane as an iceberg, which is either floating or it is embedded in the sea of lipids. Yes. Moving further, let's talk about first about lipids. So in today's video, we'll be focusing more on lipids. So lipids, the lipid are nothing but they are lipid bilayer, it is there. And this lipid bilayer is made up of phospholipids. Yes, here we're talking about the phosphor, which means the element phosphorus. This is how the actual cell membrane structure look like. Don't worry, we'll be discussing about this in detail too. So what we can see here, we can see a round thing, and then we can see the two sticks. The round thing is called as head, whereas the sticks, which are two, they're nothing but they are the tail. The head is hydrophilic or it is polar. Now what is hydrophilic? Let's just quickly divide this two terms. Hydro means water. Philic means it is having a love, or it is attraction, or it is affinity. So these phospholipid, the upper head part, is having affinity, it is having an attraction, it will attract water. Whereas the tail is hydrophobic or it is non-polar. Yes, students. Now what does hydrophobic means? Now we saw that hydro means water. Phobic means it is having a fear or it is repulsive. So hydrophilic, the head was attracting water, the tail is repelling water. He don't love water, they hate water. So it is also called as non-polar end. Moving further, and therefore these phospholipids, as one of the part, the head part is hydrophilic and the tail part is hydrophobic, and such molecules in chemistry are called as amphipathic. So phospholipid, out of which the cell membrane is made up of, is also termed as amphipathic molecules. Moving further, now what is the function and the role of the tail? It is hydrophobic, so it allows maximum resistance, which means it does not allow water soluble molecule to transport from outside to inside. That is, if I call this part as the extrinsic, which is the outer side, the water molecule cannot easily go through because the tail part is hydrophobic. Unnecessary passing of any molecules and water molecules sometimes are not good for the cell. So the tail part completely take care of that. And therefore we say that the cell membrane is selectively permeable. Very important term to remember. Okay, now one more part which is very important to understand, that the tail has some unsaturated hydrocarbon. Yes, and they are also termed as fatty acids. Okay, now what are unsaturated hydrocarbons? If we see the basic in chemistry, they are nothing but they are fats which are liquid at room temperature. So moving further, now till you must have understood the role and the function and how does morphologically the lipids and the phospholipids look like. Now we will be seeing in details why the name was given as fluid mosaic model. The way this particular arrangement of the head and the tail is like lateral. It is like one by one by one that got compared with the mosaic tile structure. So we can see here there are small pieces here of tile. So these small pieces that actually make up a tile. So these small pieces of tiles which are making the whole tile is nothing but the small head here. Okay, so the first name it got as mosaic, Itka mosaic, and the tail has the unsaturated fatty acid. Sorry, so they provide the fluidity. It allows the tail, it allows the lipid to make it more flexible. Okay, now it is not completely fluid, it allows it to make more flexible. Okay, and therefore because of the presence of UFA, that is unsaturated fatty acid, the tail is fluid and the structure looks like mosaic, and therefore the name was given as fluid mosaic model. Yes, students. Now moving further, now you'll be having a question that the cell membrane, is it completely fluid in nature? As already mentioned, no, it is not completely fluid. It is also tough. Okay, it is also having some strength, and therefore there is a specific term for the nature of the fluidity of the cell membrane, the lipids. And these term, where it is a partially fluid and a partially tough, it is called as quasi fluid. Now moving further to the next question, so how can we actually measure the fluidity of the lipids which are present within the cell membrane? This can be simply measured by the movement of the proteins within the cell membrane. So obviously you will be having a question that, but how does the protein travel within the membrane? So we will have a quick look on it. So let's see here. We have the cell membrane. Now this is the protein molecule. The protein molecule move laterally. It is moving laterally. Okay, it is moving laterally. So therefore, the protein molecule within the cell membrane undergo lateral movement. Perfect. Now, we said that there is also presence of one particular molecule, okay, which is not good for the health, but it is very essential to be consumed. Yes, and that is nothing but the additional to the cell membrane, and that is cholesterol. Yes, majority of the cell membrane, they have the small molecules in between, and that molecule is of cholesterol. Now the cholesterol actually provide the rigidity to the cell membrane. So as we said that not all the nature, not all the lipids which are there, which are fluid, the rigidity is being provided by cholesterol. Now students, as I mentioned that there are some of the differences in the prokaryotic cell and in the eukaryotic cell. The cell membrane of both the cell are exactly same, but there are some of the minor differences. We'll be jumping on that. Okay, and that minor differences is nothing but it is the cholesterol molecule. So let's move further with the differences in the prokaryotic cell and the eukaryotic cell. Okay, so in the prokaryotic cell, there is no cholesterol. Okay, so prokaryotic cell, if there is no cholesterol, if there is nothing, no. There is a molecule which looks like cholesterol, and that is called as hopanoids, or it is also called as the pentacyclic cholesterols. You don't have to actually remember the whole structure of it, you have to just remember the name. Whereas in the eukaryotic cell, we do have cholesterols, as we just saw. So I hope that you understood the concept of fluid mosaic model. Okay, if you have any doubt, then comment in the comment section. I will love to actually respond to all the doubts and the query you have. And if you have any suggestion on which topic I should upload a video, then all the suggestions are welcome. So I hope you enjoy this video. Here we come to an end for our today's video. Okay, if you like the video, then please hit the like button and do subscribe to our channel. We would love to upload more such content, and that will give us more motivation to upload such kind of videos. Okay, so we will be coming back with more such interesting topics and video. So till then, take care, stay safe at home, and see you soon.